Vlad, do you want to say any welcoming words as far as the first event of the new year?
Sure, yeah. So, yeah, in fact, so this is our first seminar in this full semester. So we're very happy to restart the seminars. We originally planned to have them in person, but I think we will leave it to the next semester, hopefully. And so we're very happy to see everybody coming back to campus and restarting usual academic year. And we are happy to restart our seminars with that as well.
Excellent. Vlad, thank you. Let's go ahead and get started. My name is Brendan McGinty, and I'm part of the Center for AI Innovation within NCSA, the National Center for Supercomputing Applications at Illinois. So happy to get things started this academic year with our first speaker, Dr. Becky Smith. Just as a reminder for the audience, this is being recorded. It will be up on our website. Questions can go in the chat, and I'll remind folks of that as we get going with a couple of chat messages, just to remind that the questions can go in there. I'll ask those questions at the end, time permitting. Becky has promised a time for that, and we may even wrap a couple minutes early. But let me go ahead and introduce our guest speaker for the day, and then get out of the way and let her get started. Rebecca Smith is an epidemiologist with a focus on decision-making from messy and incomplete data using mathematical and statistical modeling. Her home at UIUC is in the College of Veterinary Medicine, but she has affiliations at the Carl R. Woese Institute for Genomic Biology in infection genomics for One Health theme, at NCSA, and at the Carl Illinois College of Medicine. She works on a variety of topics, from zoonotic tuberculosis, vector-borne diseases, menopause, and lately, COVID-19. Becky, it's great to have you with us. And by the way, congratulations on the Illinois Presidential Medallion for COVID response leadership. What an honor, and it's an honor to have you here today. Go ahead and do the share screen, and we're just so happy to have you with us. Take it away.
Thank you, and I'm happy to be here. I have to admit that this may be a slightly different talk than this group is used to, because I'm going to be talking less about the way that we build the models and more about why we build the models, and giving some examples of how we actually build models to make this operational impact. So the first thing I'm going to talk about is just what I mean by operational impact. Now, in public health, the idea is that you bring in data, you build models, and then those models inspire some action. In academic modeling, though, too often we bring in data, we build models, and then we get a publication, and action is left to somebody else to do. I don't like that approach. I like to know that my models are actually having an action as well as resulting in publications. So I want to talk about some of the theory of how you even approach building models so that actions can be taken. And the first thing you have to ask is, what action do you actually expect? Operational actions can be on different temporal timelines, either slow or fast. They can be at different spatial or population scales, either broad, statewide, national scales, or very narrow individual response or local response. So you have to decide where you're going to be. If you want to make really broad changes in policy, you're thinking about long-term slow impact. If you want to make kind of more narrow changes, but for the long term, you're thinking about plans at the local level. If you want to make really broad impact, but fast, what you're talking about is communications and improving communications. And if you want really fast response at a very narrow scope, what you're really looking at is kind of this reactive action of, there's an outbreak, how am I going to respond? all of those require different models. So for these policy changes, we're really looking at scenario analysis, looking at what would happen if we have different policies in place so that we can tell the policymakers, the decision makers, here's what we predict would happen under these different scenarios so that they can make the decisions on which scenario produces the results that they most want for the cost that they can most live with. When we're talking about planning, we want these long-term predictions. So rather than the immediate what's going to happen next week, we're thinking about more what's going to happen in the next month or in the next year or over a 5-year planning period. When we're talking about communications, we want short-term predictions. What's going to happen this week, next week, this summer, so that we can communicate that out quickly and have people have the information they need to make their own decisions. And then when we're talking about outbreaks, we're often looking for triggers. We're looking for what will happen next week if I make a change right now. So all of those require these different modeling approaches, different timescales, and different data sets to feed into them. So for example, I realized that I said I would talk about zoonotic tuberculosis. I'm just going to use it as an example of how we might use these different modeling approaches. So for instance, for zoonotic tuberculosis policy, we're building these metapopulation models. Zoonotic tuberculosis, by the way, for those who don't know, is a relative, it's a cousin of tuberculosis in people, and it does infect people. In high-income countries like the US, we've pretty much eradicated it as a public health concern, but we're still trying to control it in some of our cattle, especially from wildlife spread or spread from imports. So when we build these metapopulation models, we're building models of the individual farms, but they're connected through an animal movement network. They may be connected through a wildlife population. And we're looking at surveillance protocols. Where and when should testing take place? Which tests should be used? That sort of policy from the USDA perspective of how to keep it under control. If we're looking at making plans on an individual farm level, we might use a risk analysis model to ask about biosecurity. Okay, you don't have TB right now. How do we keep it out? Do we need better fences, or do we need to use intake tests? So we're building this individual farm level model and looking at risk. Now, for a communication standpoint, we might want to do a risk factor analysis, and that's where the actual statistics and AI could come in, looking at what are the characteristics of farms that are experiencing outbreaks so that we can tell farms, okay, are you a high risk or low risk farm based on what we've seen are the characteristics of farms that are high risk. And then for the immediate reactive action, and this is where most of my work in TB has come, is these compartment models for within a farm, how to respond when we do find an outbreak. We find a farm that has TB on it. Should we depopulate it or not? With farms, you can actually just go in and say, okay, we're going to kill all the cows and make you start over because you have an outbreak. But that's maybe not the best choice. And obviously it's not a great humane choice, but also thinking about when to end a response. If we're going to test and eradicate, when do we declare the outbreak over? This can be solved with compartment models. So you see, those are all different types of models on the same issue, but depending on what kind of response we want to have from an operational standpoint. So let's talk first about this action response, this short-term, narrow scope, regional or local response to create action in the case of an outbreak. And I'm going to be talking about West Nile virus. West Nile virus is a mosquito-borne infection. We have it in Illinois. We are actually one of the states with the highest case numbers in the country. And especially in the Chicago area, we have to have a lot of response. Now, this brand new paper that just came out in PLOS Neglected Tropical Diseases, we looked at where actual decision responses are made. Here on the x-axis is the spatial resolution, and on the y-axis it's the temporal resolution. And you can see that a lot of the responses that they have are up here at this very local scale, daily to hourly, maybe even to weekly response, or in this light blue box, larvicide and adulticide. This is killing the mosquitoes either in their larval form or their adult form. And this is very much on the weekly or more frequent response level and at the very small spatial scale. Well, we compared that to where existing West Nile virus models are, and you can see in this daily to hourly space where a lot of the responses are done at the operational level, there are no models at that temporal resolution. And there are very few models at this local and point scale. Most models are working at a county or township, a much broader scale. Now, luckily, this orange box that I've highlighted here, that's the model that my lab works with. We call it the Chicago model because it was developed for the Chicago area. And it is on a local and point scale, but we're really working on a weekly basis. We haven't gotten it down to daily and hourly. And to be perfectly honest, the reason we haven't is that the data are just not there. But how do we actually do this? How do we break it down to this smaller scale? And this is work that I'm going to present that was done by Johnny Ullman for his PhD. He's now a postdoc at University of Michigan modeling COVID and flu. But for his PhD, he worked on West Nile virus, and he looked at Cook and DuPage County. So this is Cook and DuPage County here. The dark blue is the Chicago city limits. And he looked at this model that Surendra Karki, a previous PhD student in the department, had built on these 1 kilometer hexagon levels. So he's trying to predict within each of these 1 kilometer hexagons, what is the risk for human cases. And you can see here that these are filled by the number of human cases. This is very sparse data. There are not a lot of human cases. Chicago is one of the worst areas in the country, but it's still not a huge number of cases given the population. And trying to predict using biological and environmental and weather, but also some anthropogenic data, trying to collect as much data as possible, including this field season here. It looks very simple. Okay, field season. This was Johnny actually going out and sitting in these hexagons and collecting by hand any mosquitoes that landed on his body to try to find out if that could give us more information to predict these cases better. So what exactly can we use at these very fine scales to predict cases? And he developed this, he calls it the bang for the buck performance covariate or covariate performance index. So what he did was look at how important was the covariate to his final model when he had all of this data from all of these different approaches compared to these categories of how difficult was it to collect. So category 1 was precipitation, temperature, and year. Those are very easy data to collect. Category 2 is things that you can get from the census or from just simple analysis of the precipitation and temperature data. Category 3 required mosquito trapping. It required doing a lot more working with the, he was tracing out building perimeters, finding out how much space there was in buildings. So it took a little bit more work. And then level 4, these shaded ones, those are him actually going out and standing and being a, we call it human landing catch. So he was a human mosquito trapping machine, and some other work that involved really fine scale work with the satellite data. And what he found was that most of the really important covariates didn't take as much work to collect, which is really important when we're thinking about from an operational perspective. Are we going to respond or have the data to respond and say, okay, you need to go out and trap, you need to go out and spray for mosquitoes next week? It's much easier if they're easier to collect data. So this is our use of modeling to try to decide which data do you actually need to collect, because in order to collect it on the scale that we need for these quick responses, you really have to have it be easy to pull together quickly. Part of our problem though with the mosquitoes is all of that. If you go back to here, this level 3, this all requires mosquito trapping. And we have a sampling bias problem with the mosquito trapping. This is work done by a current student in the program for ecology, evolution, and conservation biology, Sulagna Chakraborty. And we were looking at mosquito collection and testing across the state. And my lab has a data use agreement with the State Department of Public Health. So we have all of the mosquito trapping data from the entire state. And here is a map looking at the state by county and their mosquito or minimum infection rate, which is a measure of how much infection is in mosquitoes. And you can see that it varies across the state. This is a 3-week period in 2016, which was a really bad year mosquito-wise. And it looks like, okay, we just have some variation. But if you actually put on top of that the number of traps that were used to collect that data, you can see that it's actually quite variable how much data went into those numbers at the county level. So for instance, this county had 0 mosquito infections, but they were sampling a lot of mosquitoes. So they had a lot of mosquito pools, a lot of traps. They had good coverage of the area. That is a 0 I can believe. Whereas here, there's an entire 7-county area that did no trapping for mosquitoes in the middle of West Nile virus season in one of the worst years that we've had recently. So those are 0s that don't really mean anything because we don't know what was out there. Likewise, up here, Cook and DuPage County, we look at that very high infection rate and we say, yeah, I believe that because they do a lot of sampling. Whereas over here, we have a very high infection rate. Well, that's just 1 trap. And it happens that they had very few mosquitoes in that trap. So it looks artificially large. And that plays a role in how much we can trust the data and what kind of response we can make to it. Sulagna's work was trying to decide, what does it mean that we have these differing amounts of sampling in these areas? And she looked at how much error comes into our estimation if we sampled less. And for this, we used an economics approach called value of information, where we took the data from Cook and DuPage County, which was, remember, just covered in traps. And we said, okay, well, what if we just randomly take some of those away? And so we estimated the infection rate at different densities of trapping and then compared it to the observed using 100% of the traps across a 9-year period using all weeks of data, both counties. And what we came up with was that, yes, as trap density decreases, we have a really big impact on how much error we have. The fewer traps you have, the more error you're going to have in your estimation. So the less useful that will be for making an operational decision, because if you have a 0, it becomes much and much less useful at deciding, is that a real 0? But importantly, we also found out that as the infection rate went down, so when infection was very low, usually at the beginning of the season, when you're just starting to see the infected mosquitoes, that also played a role in our error. So a low infection rate was synergistically bad as far as our ability to estimate this value that we use for making operational decisions. So at the very beginning of the season, when a lot of people say, I'm not going to put out a lot of traps because the infection rates are low. Well, no, that's when you need the most traps. And so we were able to give this advice to our mosquito abatement district partners that actually when the infection rate is low is when they need the traps the most to be able to detect it early and know when there's going to be cases coming, when they need to respond. So what does that mean for Cook and DuPage County? Well, these black lines In the middle here are the observed values, and the red lines are estimated error around it. Doesn't mean much for them. They do a lot of trapping. But if we use these other counties where they don't do as much trapping, we can see how much error we estimate to be around their estimates of the infection rate. That's really going to make a difference if they only go out and larvicide or adulticide when the infection rate gets above a certain point. Some of these areas in here, they may be underestimating by a lot how much infection they have in their mosquitoes. And we also had some work looking at actually what is the impact of adulticide. And this is work that Dr. Danielle Sass did as part of her PhD in the Department of Statistics. So here is the Northwest Mosquito Abatement District. It's one of a number of mosquito abatement districts in the Chicago area. And the red and the blue points are the different mosquito, or the red and the yellow points are the different mosquito traps. The blue points are where the weather stations are, so she can get the weather information. And this is the distribution here of the different values, the number of mosquitoes or the abundance, the infection rate, and what we call the vector index, which is the product of abundance and infection rate. And looking at a daily and weekly value, you can see that they're very skewed data. They're highly dependent on weather. So the mosquito abatement districts asked us, can you tell us, are our sprays, when we go around with a truck and spray for adults, are they actually having an impact? Well, Danielle used a lot of really great advanced statistical approaches to look at, do they impact abundance, infection rate, or a vector index? And what you'll see is that the abundance is actually decreased greatly. So blue would be lower than expected and red would be higher than expected if using just the weather. So for the first couple of days, you do see a big decrease in the number of mosquitoes. Not a big difference, though, in infection and vector index. And once you get 6 days out, you actually see what we call the bounce back effect. So the trucks go out and they spray and they kill all the adults that are there. And then you've created this open space for any emerging adults that they come out, and you get more adults ending up a week after you spray than there would have been if you hadn't sprayed, based on our models. This is very useful from the operational perspective so that they can know, okay, spraying once isn't effective. And they now have trials going on to see, well, what if we spray twice in a week? Would that block out this bounce back effect? But they can't plan those experiments until they have proof that those are needed. So that's how our modeling is supporting our operational partners on the ground in Chicago dealing with West Nile virus. We also had a project with NCSA trying to make all of these modeling available. And this was work done by Marcus Salavinas and Max Ammon. They did a lot of work on it. It's paused at the moment because our funding ran out, but hopefully we'll be able to get back to it. And it's a way for areas to be able to upload their data. So if we don't have their data, they can upload it themselves. And then it will, for the county, use our model to predict out what's going to happen in the next couple of weeks as far as the mosquito infections. And so this is a very useful tool that the mosquito abatement districts love this, that they have the use of our models to predict what's going to happen based on what we get from the weather and what we've been seeing so far. And so they can make changes for the next week or two, this local and high-speed response based on our modeling approaches. So that was just talking about this narrow local scale fast response. What about broad scale fast response? And that's really the communication aspect of how do we reach out to people and let them know that something's going on and use our models to help from that aspect. And on communication, we use a lot of this on ticks and tick-borne disease, mostly because personal responsibility is about the only thing we can do to prevent tick-borne disease, because there's no way to effectively block ticks in the environment, unfortunately. So a lot of what we've been doing is trying to improve our surveillance. So this is a map of 3 different species of ticks in the state of Illinois. This is the Lone Star tick, the American dog tick, and the black-legged or deer tick that most of you have heard about because of Lyme disease. Now, the filled in spaces, the orange and yellow, are what historically we knew about the ticks in the state of Illinois. And then we've been improving our surveillance and trying to use models to predict where we would expect the ticks to be and when we would expect them to be there, to go out and look for them and try to fill in these blank spaces on the map. Because we still have a lot of blank spaces on the map. And if we don't know the ticks are there, we can't warn people that they're there and help them protect themselves. And what we hear a lot from people, especially in Southern Illinois, is that when they feel like they've been exposed to a tick-borne disease and they're having symptoms, they have a hard time getting treatment or diagnosis because the medical professionals say, well, we don't have those ticks here. Well, as you see from this cross-hatching, we're finding out that there are black-legged ticks in way more counties in Southern Illinois than we ever knew before. So we're using these model-based surveillance to try to improve our understanding so that we can communicate that better, which will then improve diagnostics. We also have been doing a lot of surveys. This is a COVID-safe activity. So when we couldn't go out into the field, we started these knowledge, attitudes, and practices surveys. And this is results from the survey of veterinary professionals. We asked about their knowledge about ticks. So this is looking at their knowledge score on the y-axis, and on the x-axis is how many years since they last had a tick training. And either overall or specific to the diseases or specific to the ticks, we see that if the longer it's been since they've had any training, this is those who have had no tick-borne disease training, then the worse their knowledge score is. Okay, so we need to get training out there. We need to start telling these professionals about ticks and tick-borne disease. And by using this approach of finding out what's out there, yeah, we need to do training. And when we asked them if they'd like to have training, most said yes. No, they didn't agree on what kind of training they want. So we're gonna work on that, probably do a little bit of everything, but they all said that more outreach would be better because they can't respond at the local level unless they know what's out there. So we're using our modeling approaches to give more information on ticks and tick-borne diseases in these areas around the state. We also are doing outreach for, we found that there are certain communities that just weren't being reached about tick-borne disease. So we do outreach. This is an infographic that Heather Costco, a postdoc in my lab, had developed for hunters. We found that, yeah, hunters are at really high risk for tick-borne disease, but they're not necessarily protecting themselves. So she put together this to tell them what they can do, because we know that the areas that are really popular with hunters at the time that hunters are out there are also really good tick feeding areas. Okay, so those are the quick response things where we're talking about in the next couple months or the next couple of weeks. But what about the slow changes, making plans and making policies? On the planning support, this is a project that was, again, part of our collaboration with NCSA, and especially Chen Wang did an amazing job on this. And this is we called it the SHIELD K-12 Playbook. And I'll say that the slides were from Chen as well. So thank you, Chen, for the slides. The idea was that schools are not experts at controlling infectious disease. They're schools, they're experts at educating our children. So we used what's called a decision tree model to develop this playbook that helps walk K-12 administrators through the process of designing a COVID control plan. So at the back end, it's a decision tree model that based on one decision, you might be asked other questions. and it walks you all through it. But from their perspective, they're just being asked questions, and it's a multiple choice test as far as they're concerned. So they see we have a number of different modules. We've added more since this screenshot was taken, and they'll have a question like, who will be doing cleaning? And they get to select who is going to be doing the cleaning, and then that will ask them further questions along the way. It helps walk them through the decision process of how to do these controls. And then at the end, they get their decision report, which is, here's all the questions you were asked and what you answered, and a checklist where we say, here are the things you need to do to actually implement your plan. Here's all of the things you need to check off on your checklist. And here's some resources, links to resources to help you do each of those steps. And the idea behind this was just to take the modeling decision approach and make it much more useful to people who that's not their expertise. Also, been working greatly with NCSA on policy support for COVID. You may remember last year, we were sending out monthly risk surveys where we asked people, what are you doing? And what activities are you doing? How do you feel about COVID? Well, a team at NCSA, led by Wei Hao Gay, was doing a lot of the analysis, paired those risk surveys with our test data. We could pair them behind the scenes with our testing data to look at the time to a positive test based on all of these time-dependent covariates of, well, you said this month that you're wearing a mask everywhere, but now this month you say you're wearing a mask almost everywhere, so that we could look at what are the different risky activities. And part of this is for this communication. So we asked students, what activities do you think people are interested in doing? The one that shocked us, of course, is that they said, oh, sharing drinks. Who shares a drink during a pandemic? But they wanted to know, is it risky? And we could find out using this modeling approach, which are the riskier activities and which are the safer activities. But for a policy standpoint, there was a question of, should we move classes online? And this has been a question, it's a continuing question. And one thing they found was that attending class is not a risk. So this is what we call a Kaplan-Meier plot for those who aren't familiar with survival analysis. On the y-axis is time to event, in this case, testing positive for COVID-19. And on the x-axis is the timeline. So over time, people who did not attend in-person class back in the fall of 2020 tested positive faster than people who did attend in-person class. So not only was attending class not a risk, it was associated with actually safer activities. So we could say, yes, we have evidence that this policy of having in-person classes is safer. So what was actually risky? Not wearing masks, having a large social bubble, saying you interact with a lot of people, and attending gatherings, which are all the things that we knew were risky, but now we had proof using our modeling approaches to back that up. We also use this for vaccine planning on campus. When we asked people, do you plan to get vaccinated? 90% said they would, most likely. And most of those got vaccinated quickly once vaccines became available. So the people in the fall who said that, yes, when a vaccine becomes available, I will get it, by the end of the spring semester, most of those had gotten it. But that 10% who said, no, I'm not going to get vaccinated, were more likely to say, no, I don't wear masks, indoors or out. And they also were associated with larger social bubbles, which, remember, we found using this risk survey approach were really high risk for infection. So using this data and the modeling approaches, just looking at the data, we could say that that 10% of people who would not get vaccinated on their own were high risk for infection. And that helped to make the decisions on campus to require vaccines and now to mandate vaccines. Moving to another study that we did with COVID for policy decisions was the question of how frequently to test. And this is using the COVID Detect study, which was run on campus, also at Northwestern University. We paired with them for this, but I'm only going to share data from our campus version of the study. So the idea of COVID Detect was to enroll people within 24 hours of a positive or within 5 days of a close contact, and only if they'd had a negative test in the last 7 days. So we're finding infections really early in the process. And every day we took saliva for PCR, we took 1 nasal swab for PCR and culture, we took 1 nasal swab for an antigen test, and we gave them a symptom survey. And I'm only going to talk about the comparison of the testing here, but the symptom data are fascinating. But anyways, for the policy perspective, and this is a question that NIH brought to us, and CDC was behind it as well. They wanted to know, how frequently should we use antigen tests to detect infections? If we're going to do serial screening with antigen tests, what frequency do we use? Now, here on the right is the data on daily sensitivity. So remember, these are all infected people. So how well do the tests detect these infected people? And we align them by the first viral culture. So the viral culture is kind of our proxy for infectiousness. So the idea is at day 0, this is their first day of being infectious. And you can see that the dynamics over time, prior to that first day, the saliva was actually a bit better than even the nasal PCR at detecting those cases. The antigen tests were not really good at finding people early in infection. And as time went on after that viral culture, the saliva PCR actually dropped away in sensitivity, meaning that people became negative faster after that infectious period. And the antigen even faster, whereas the nasal PCR, this has been a known problem that the nasal PCR stays positive for a really long time. But the question is, can we find the infected, and can we find them before the infection spreads? Now, if we break that up to just those 3 periods, before viral culture positive here, while viral culture positive here, and then after the viral culture positive here, we see that the antigen is really good at finding them when their viral culture is positive, but before and after, not so much. Whereas the 2 different PCRs are not as great at finding it before positive as when it is positive, but they still retain that positivity after the viral culture goes negative. So we used, again, the value of information approach, that we look through the different testing protocols. We had 14 days, up to 14 days of data on each person. And we said, well, what if we tested them every day? Would we find them? What if we tested them every other day? And we went through all of those protocols and looked at, could we find them at all? So that's whether or not we could find them, this top number. And then could we find them while they were still infectious or before they were infectious? This is down here. So before that culture positivity ended. And you'll see here that if you test every day or every other day or every 3rd day, you are really good at finding those infections. But once you hit every 4th day, then that sensitivity starts to go down. And that was used to drive policy from CDC and NIH. So now when you see them recommending serial antigen tests, they are recommending that you use them twice a week. Why? Because of this model right here that showed that if you started doing less frequently than every 4th day, your ability to detect infections would go down. And if you want to find these people while they are still spreading the infection, beyond every 4th day, you're below 75% ability to do that. So this entire study was driven by a policy question, and the analysis was driven by a policy question. We're doing other things with the data. They're beautiful data. But the whole question was, can we determine what policy will best find the infection before it spreads? And we did. We said, you want to test twice a week. All of these data are pre-Delta. So when we look at these and this frequency, this is pre-Delta. We know Delta becomes infectious much faster, which is why now we are doing every other day testing on campus rather than every fourth day. So going back to that policy question, or just the overall question of what action are you trying to create? Are you trying to change a policy, or are you trying to respond to an outbreak that makes a difference in what model you're going to use, what approach you're going to use, what data you're going to use behind it? I have to thank all of the people I've worked with. Obviously, I didn't go into the details on any of these models, but there's an incredible amount of data and work and modeling behind all of this, and all of the funders on the COVID. It's the COVID-19 Test Us group and the RADx Tech, Rockefeller Foundation and Shield, Illinois. On the vectors, a lot of it is Midwest Center of Excellence in Vector-Borne Disease and the Socio-Ecological Synthesis Center, which funded a lot of the work on modeling. And thank you for listening. I'm sorry if you were hoping for a lot more details on the models, but I think it's really important to approach it from a philosophical perspective before we get into the nitty-gritty. So thank you for listening, and I'm happy to take questions.
Becky, that was fantastic. What a fascinating talk. I considered it very interesting. Data-driven, modeling-based policy decisions. I'm relieved to know that the CDC and NIH are considering real data when considering policy, and that affects us all. Very good. I have a question in the chat from Zhaozha Liao, and Zhaozha leads our data analytics team for our industry program at NCSA. She has a couple of questions. The first one is, what are the statistics models or machine learning models, for example, linear regression or random forestry, that have been tried with this research?
So that depends on which research question we're talking about.
Right, it was a little earlier in the presentation.
Yeah, so for the West Nile virus modeling, within my lab, Johnny Ullman's work was mostly working on logistic regression or Poisson regression, looking for case numbers. For some of that, though, we have attempted some random forest approaches, and actually Wei Haogei's group at NCSA has been working on some random forest approaches. The problem is that sparse data and the sparse case numbers, and this is a problem that we have with West Nile virus modeling everywhere. And this group that published that paper that was talking about modeling and operational responses, we have all the West Nile virus modelers in the country in that group. It's great to get together and talk. And we all have this problem that we're really good at predicting where cases won't be, but very few people can actually predict where cases will be because we have so few cases to go off of. So random forest has struggled, although there are random forest models out there, but they do struggle to predict where the cases are going to be. But like I said, we're all really good at ruling out where cases are not likely. So at least that lets us focus in on where, well, the model hasn't ruled it out.
You just went to where her second question was, which was, how do you deal with very sparse data during the training? So to that point.
Yeah, and I'm hoping my research moving forward, once we get over the COVID hump, I really want to work on methods of bringing error propagation through from our data, from the sparse data, propagating that error through to our estimates so that we're giving a little bit more reasonable estimates, understanding the sparseness of our data. So if people are interested in that, let me know, because I'd love to collaborate on some issues around error propagation through our modeling approaches.
You know, earlier in your presentation, you had a map of the state and you showed some of the white counties where you just can't get enough data, right? And I noticed, how did you put it? You said, you know, the human collector, right, to trap mosquito data, or how it seems laborious. It seems that is there technology that you can leverage to get more data and sensor data, anything like that you're employing?
Yeah, so those human landing catches, the CDC does not like them, I should say. CDC does not like it that Johnny was out there. He did that on his own because he really thought that it was a good way to get more information. Now, we have these traps that we can put out. So you put out a trap and one night later or 2 nights later, you come out and you collect all the mosquitoes from the trap. That's mostly how we're doing it. But we have to remember that in some areas of the state, vector surveillance is mostly done by public health departments. And in some areas of the state, one public health department might cover 5 counties, 5 large counties. And the person responsible for mosquito surveillance is also responsible for tick surveillance, also responsible for food inspection, also responsible for pollution investigations. I mean, it's one person doing all of this. So the reason we don't have data is not that they don't care, it's that they have too much going on. And we have done actually surveys of people with regards to both mosquito surveillance and tick surveillance. And what they tell us is they know they should do more, but they don't have the people and they don't have the budget. And without the budget to hire the people, that's not going to change. So the surveillance is going to have to work with less data.
A common problem that doesn't get us to the ideal, does it?
No.
I'm wondering if there are other questions, please feel free to either raise an electronic hand, put it in chat, speak up. Happy to take other questions. Give people a chance here.
Yeah, so I have a question. You know, in the very beginning, you mentioned that, you know, in academia, we very frequently collect data, develop model and publish a paper and move on. And you wanted actually to build this to be actionable models. So what are the biggest sort of hurdles in making models to be actionable or useful beyond publication?
So some of the biggest hurdles is actually on the communication front. So working with the people who would actually be using the model so that they understand what the models can and can't do. So we're lucky with the mosquitoes, the mosquito abatement districts, they're amazing partners. They have PhDs, the people who run the districts, they have PhDs, they're entomologists, they understand modeling from a certain standpoint. But when we're talking about other approaches we're doing, you know, my work on zoonotic tuberculosis, a lot of that work now is in Ethiopia, where the Ethiopian dairy farmers who have 5 cows and they sell milk at the farm gate, they don't understand what the model is saying. They just understand you're coming in and telling me that cow has tuberculosis and is going to make me sick, but that's my income. So thinking about working with the people who understand the models or communicating the results of the models in a way that is useful to people. And we tend to in academia say we have to be more specific. We have to get finer grain. We have to deal with all the errors. We want really the best model possible. But if we can't actually translate that to something that people on the ground can use, it's not going to be used in actually making action. So a lot of my work is thinking about what the people on the ground need and designing the model to answer that need, rather than designing the model that from a scientific perspective might be most interesting to me.
Yeah, that's actually a very interesting question. Answer, because, you know, we very frequently don't think about that. We just think how we can get the best possible model prediction value or something. But what does it mean? Is it, what do you do with this? Is this useful in any way other than listing it in some sort of paper or plotting on a graph? Yeah.
Yeah, and this is why I highly recommend multidisciplinary teams. So my approach to this is because I'm a veterinarian first. I fell into modeling halfway through vet school. But my plan, I was going to be in dairy practice. I was going to be out on dairy farms. So my perspective is always, I want to be out there on the farms helping the farmers. And that just kind of set up my approach to things. Having these multidisciplinary teams where you have the people with the mathematical and statistical expertise, pairing with people like me who are closer to the field, pairing with the people who are actually in the field, that's really necessary to get these really impactful models built.
You know, Becky, in our world, we talk about multidisciplinary a lot, and it really struck me how truly multidisciplinary across colleges you are. How did you make that happen? I assume it was the old-fashioned way.
So I'm a big proponent of a liberal arts education. I'm a graduate of a small liberal arts college where, I was taking classes with people in all sorts of fields, and then that started it. And then we've always just, throughout my training, the focus was try to get the data that we need and try to get build the model that we need in the field. And to do that, we needed to be multidisciplinary. So we would reach out to the computer scientists and the physicists when we had mathematical problems we couldn't solve, but we're reaching out to the economists when we have questions about the economics that we couldn't solve. Because when the farmers come to us and give us a question, or when the mosquito abatement district comes to us and gives us a question, we're not going to say, no, I don't do that. We're going to say, who does do that? And how do we make it work? So for example, the question on what effect does the spraying have? Danielle Sass was a PhD student in statistics. When the mosquito abatement district said, that's the answer we want to know what this does, I said, I can't do that, but I know Dr. Bo Lee from the Department of Statistics. And so I reached out to her, and she found me a student who was willing to work on the project.
Outstanding. Other questions? Well, seeing none, Dr. Rebecca Smith, Becky, thank you so much for your time today. The perspective of all of this, VetMed, the interdisciplinary aspect, the data-driven research. I guess one last quick question. AI across those disciplines. We're an AI center here, and it feels like there is a growth of at least the understanding of how to leverage AI intelligence in some of the research. Are you feeling that? Are you seeing that?
Yeah, so since I'm a practical person, my approach is, if AI can do something, then use the AI, and the AI can certainly do things that we can't do with traditional statistics, but use the approach that works. So I do compartment modeling, and I do AI, and I do traditional logistic regression. So whichever approach works, use it. But absolutely, there is a growth of areas where AI can really be helpful to us, especially network modeling, I think, is going to be of most interest in epidemiology, because most of what we're working on is highly interconnected network type situations.
Excellent and ridiculously practical. Thank you so much for being here. Thank you for your time. Thank you everyone for joining us again. This presentation will be on our website in a few days. Really appreciate your time. Looking forward to seeing you next week. Have a great rest of your day. Thank you.